Assalamualaikum warahmatullahi wabarakatuh. This time our group will present the result of our group's work about transport of amino acid through the cell membrane. First of all, let's take a look at all the components in the cell membrane. The cell membrane itself is made of phospholipid. As seen here, a phospholipid has a hydrophilic head and two hydrophilic tails, thus making multiple phospholipid molecules to come together and form a bilayer. This structure is called flat mosaic, meaning that phospholipids are free to move around but they stay together as surf. This can pass through the bilayer easily if they are soluble in lipids. In the phospholipid of the bilayer, there are cholesterol molecules which stabilize the bilayer by keeping phospholipid at relatively normal distance from each other. When it is too cold, cholesterol keep the phospholipid apart, so it's still fluid. When it's so hot, cholesterol keep the phospholipid together so the bilayer doesn't fall apart. And then we have glycolipid. Glycolipid are carbohydrate molecule attached to the phospholipid, hence the name glyco and lipid. Glycolipid act a identifier of the cell and the carbohydrate molecule are recognizable by other cells. For example, when a phagocytosis sends a glycolipid that is specific to bacterium, it will be triggered to destroy that bacterium immediately.
Cell transport is one of the important functions of the plasma membrane. In general, the transport mechanism of the cell membrane is divided into two, namely passive transport and active transport. Passive transport is the transfer of substances or molecules from high concentration to low concentration and doesn't require energy. Examples: diffusion and osmosis. Active transport is the transfer of substances or molecules or ions from low concentration to high concentration against the concentration gradient and require energy in the form of ATP. Examples: natrium, kalium ion pumps, endocytosis, and exocytosis.
The next topic we're going to discuss is integral protein. Some molecules, such as oxygen and carbon dioxide, that lipid solubles can pass across the membrane freely. Some molecules that are not lipid soluble cannot get across the membrane because the membrane is made of lipids, which is hydrophobic. These molecules need integral proteins to help. These are transmembrane, or we all know as integral proteins. There are many molecules that need to be transported. Different molecules have different methods of transport. Some of them need energy and some of them do not. They who don't take energy transported by osmosis process, where molecules go from the higher to the lower concentration, or we can say passive transport. Integral proteins divided into two types. The first one is carrier protein, which is work both in an active transport and passive transport. The second one is channel protein that works in an active transport. The first proteins we're going to discuss is carrier proteins. So when the molecules bind to the protein, this guy changes its shape to accept the molecules and then spit it to the cytoplasm, and they transform again to the normal shape. So as Afrizal explained before, membranes are made from lipids, which means they are hydrophobic. So molecules such as water cannot get across the membrane, so they need alcoholin to transport it. And this transport don't take energy, which count as passive transport or facilitated diffusion. And the last one is ion pump. Ion pump pump specific ion from the cytoplasm to the exterior of the cell. For example, this pump over here is sodium ions pump that only pumps sodium ions. And there's also proton pumps that pumps hydrogen ions. Ions pump is also essential for the co-transport mechanism along with the carrier protein.
In this case, I will be explaining how amino acid is being transported through the cell membrane. Amino acids are the building blocks of proteins, which are building blocks of our bodies. They are essential to life because proteins play important roles, such as performing enzymatic reaction and providing structural support. We acquire proteins by taking amino acids from the proteins of other organisms. So, the digestion of amino acids required two proteins, a transport protein and an ion pump, since the transport protein does not work without a sodium ion. An ion pump is required to perform this transport. First of all, the ion pump pumps sodium ions outside the cell, creating a higher concentration of sodium ions outside the cell than the inside of the cell because of the concentration difference. The ion binds with the transport protein and changes its shape to fit an amino acid. The amino acid then binds with the transport proteins when both the sodium ion and the amino acid are in place. The transport protein changes shape once again and spits out the amino acid and the sodium ion inside of the cell. The transport is complete. The transport protein can change shape to its original form and ready to perform another transport.
Okay, next I will talk about endocytosis. Endocytosis is the process by which cells internalize substances from their external environment. It is how cells get the nutrients they need to grow and develop. Substances internalized by endocytosis include fluids, electrolytes, protein, and other macromolecules. Endocytosis is also one of the means by which white blood cells of the immune system capture and destroy potential pathogens, including bacteria and protists. The process of endocytosis can be summarized in three basic steps. Number 1, the plasma membrane folds inward or invaginates, forming a cavity that fills with extracellular fluid, dissolved molecule, food particle, foreign matter, pathogen, or other substances. Number 2, the plasma membrane folds back on itself until the ends of the infolded membrane meet. This traps the fluid inside the vesicle. In some cells, long channels also form extending from the membrane deep into the cytoplasm. And the last, the vesicle is pinched off from the membrane as the ends of the infolded membrane fuse together. The internalized vesicle is then processed by the cell.